One by one. Okay. Hey, I am Mariam.
Hi, I'm Annette.
I'm Sharbet.
Okay, our topic today is about the neuromuscular junction, and I will start. Actually, the neuromuscular junction is a chemical synthesis between a motor neuron and a muscle fiber. It allows the motor neuron to transmit a signal to the muscle fiber and causing muscle contraction. The muscle require innervation to function and even just to maintain muscle tone, avoiding atrophy. First, the motor neuron axon travels from the anterior horn for the green matter in the spinal cord to the motor end plate. Then the electrical signals travel with the neuron membrane as action potential with the release of neurotransmitter. They become chemical signals, and after the postsynaptic membrane, they become electrical signals again, and the muscle cell is depolarized, just like this.
Okay, so these neurotransmitters, mainly the acetylcholine, triggers these muscles to contract. The acetylcholine is formed from two molecules, which is the acetate and the choline. The choline is a type of nutrient that is found in extracellular fluids from food intake, and it is later absorbed by blood vessels from the gastrointestinal tract. The choline is absorbed into the nerve terminals by choline channels, and it binds with acetyl-CoA, which comes from mitochondria. It is released from the nerve soma, which is the nerve body, in the gray matter. The acetylcholine is bound by choline acetate transferase enzymes, and the acetylcholine is absorbed by acetylcholine receptors into the vesicle by its special channels. The vesicular membrane also has special proteins that binds calcium ions. Now, with action potential, there's an influx of sodium ions and calcium ions, and then into the terminal, and then there's potassium influx outside of the terminal. So this changes the internal voltage from about minus 70 millivolts to 10 millivolts and 30 millivolts when the calcium ions come in. The change in these charges inside the synapse attracts vesicles to the nerve end membrane, where the synaptobrevin protein on the vesicular membrane binds with syntaxin. The vesicles then dissolve, opening at the nerve ending, and neurotransmitters are released into the exocytosis.
Okay. After that, exocytosis takes place. The neurotransmitters are released into the synaptic cleft between the synapse of the nerve terminal and the motor end plate of the muscle cell. This neurotransmitter travels and then it's absorbed by the acetylcholine sensitive channels. They're also sensitive to nicotine because of the nature of the structures of the acetylcholine and nicotine. So these receptors, they attract acetylcholine and is triggered then to open, and happens an influx of sodium into the nerve cell, into the muscle cell in high concentration, and also potassium influx outside the cell, but it's not in as strong concentration as in the sodium. So it's little potassium influx outside the channel and a lot of sodium influx inside the channel of the muscle cell. In this ligand-gated channel in the motor end plate. So the voltage changes in the muscle cell from 80 millivolts at rest to approximately 0 millivolts then it's depolarized. And it happens continuously along the muscle cell when it transferred from the chemical signal into an electrical signal. The acetylcholine that's bound already to the sensitive channels then is broken down by an enzyme that's found on the motor end plate, and this enzyme is called acetylcholine esterase. It breaks down acetylcholine again to acetyl or acetate, acetyl-CoA, and choline to be recycled and used again. I think I'm done.
Can we show them the
Okay.
draw?
So this is how the action potential. It is communication between the nerve terminal and the muscle.
Is it showing?
Yeah, it's showing. The spinal cord, the nerve axon, synapse, muscle cell. This is the motor end plate. So this is how the action potential travels in the nerve, in the muscle cell, triggers the muscle movement with the use of acetylcholine. Hope that was very helpful, and thank you all very much.
Bye bye.